Yeah. Can you share your screen?
Yes, ma'am. Is it visible?
Um. Yeah.
Ma'am, I've typed the same command, sudo app hyphen
Okay.
get install, but
Okay.
this
So,
time,
Saima ma'am, yeah, like you have installed this Linux first time in your system.
no, ma'am, I've installed this in the class.
Okay. So
Yeah.
maybe the aptitude it is not present because the package you can see it is throwing the error. It is unable to locate. So just
Ye-
write here
yes, ma'am.
sudo
Okay, sudo
space apt
space apt
hyphen get
hyphen get.
space install
Yes, ma'am.
aptitude space aptitude.
Aptitude.
Yeah.
Okay, and this one.
Yes, enter. Enter ma'am. Hello. Can you hear me guys?
Yes, ma'am.
Okay, maybe like she is having a error in network issue. What about others? Did you find any difficulty in the introduction part?
No,
Anything
ma'am.
you want to know? Jai Ganesh sir, Lina.
No, ma'am.
Okay, fine. So, did you try to install the tools?
Yes, ma'am, did.
Okay, so it is done for all of you. Saima ma'am, I can't hear you.
Ma'am, am I audible now?
Yeah, can you share
Uh,
your screen now?
yes, ma'am. I've shared my screen. Is it visible, ma'am?
Not yet.
Yes, ma'am. Now I've shared.
Okay,
Ma'am.
enter.
Okay, ma'am.
Okay, so it is giving you error here. The aptitude has no installation. Can you just update your system? Just write here sudo apt hyphen get space update.
I'm enter.
No, no, no. Don't give a underscore there.
Okay, 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 okay.
Sudo apt hyphen get update.
Oh, okay. Sudo apt get. Yes, ma'am.
Done? Okay.
Yes, ma'am. Ma'am, so after the update, I have to type the command again for downloading
Yeah,
the software for the
yeah. You
BWA.
have no, no, no. You have to run the command for the aptitude because, ma'am, the aptitude is not installed in your system. So if the aptitude is not there in the system, that means the tools cannot be installed because the tool informations each and everything information for the tools, it is there in the aptitude only. That is the reason that I am installing the aptitude in your system.
Okay, ma'am.
Just run this sudo apt hyphen get install aptitude.
Ma'am, again I have to type the sudo apt sudo
Yeah,
apt
yeah, yeah.
get sudo apt get.
Hmm.
Then, ma'am.
Just wait. Apt hyphen get install. Give the space install aptitude.
Yes, ma'am. This.
After install, ma'am, there is a space.
mm, do you want to continue
Yes.
with?
Y. Just type Y.
Yes. Is it done, ma'am?
Yeah. Now just type here sudo. Give the space.
Ma'am, is it not visible?
No, just write here sudo
So,
space aptitude space
aptitude space.
search fast QC. Enter. Now you can see in the aptitude the fast QC is there, right? So you can install this tool now. Sudo apt hyphen get install fast QC. Enter.
Invalid
No, no,
operation
no.
found.
Ma'am, you have to write sudo apt hyphen get install. Spellings are wrong, ma'am, for install.
Yes, ma'am.
Hmm. Yeah, enter. Yes.
Ma'am, it is showing done.
Yeah. Now we need to check the FastQC tool whether it is present or not in your system or it is successfully installed or not. So for that you have to open the manual. So for that you write FastQC space hyphen help. Spelling FAST.
Yes, ma'am. Sorry. Fast QC
Yeah.
space.
Space hyphen help. So you can see enter. Yeah. The manual it is open. Can you see that? So that means FastQC is successfully installed, ma'am. Okay.
Yes, ma'am. Yes, ma'am.
Okay. So you can install the tool later on. Now let's start with the today session.
Okay, ma'am. Sure. Thank you
Yeah.
for that support.
Okay. Okay. So before we execute the pipeline, I want to tell you some important terms that you should know. So any idea what are the type of sequencing we are doing? Have you ever heard about the single end sequencing and paired end sequencing? What is a single end sequencing, guys?
Maybe one strand is sequenced.
Yeah, who's this?
Pangsha.
Oh, nice. Okay, as you know, there are two strands. Okay, for example, this is from 5 prime to 3 prime, 3 prime to 5 prime, right? There are forward strand and the reverse strand. So, for example, I'm ligating the adapter on one side, for example, on this side. Okay, and after ligating of adapters, sequencing machine, it is start reading from this end. Okay, so it will generate what? The forward file. But if the adapters are ligated on this end, because this is the reverse strand, if the adapters are ligated on this end, then the sequencing machine, it will start reading from this side and it will generate what? The reverse file. Okay, so in the single end, what happen? If the adapters is ligated only at one end, it will form forward file or reverse file. Whereas in the paired end, what is the paired end, Kansha ma'am?
Opposite strand also getting adapter attached.
Yeah, so here in the single end, what happened? The adapters are ligating at only one end, right? So that is why it is starting reading from the forward direction and it is creating a forward file. But here in the paired end, what happened? If the adapters is ligated from both the side, so it will generate what? Two files. So it will generate two output. So for example, this is a strand and I'm ligating the adapter on these two ends. So this adapter will start reading from this end and this will start reading from this direction. So this is what the paired end sequencing. Okay, so in this data, in this paired end, what happened? You are getting two files, that is the forward and reverse file. But in the single end, you are getting only one file. Okay, So here the forward file will be denoted with the ID underscore 1. So underscore 1 will indicate the forward. ID with underscore true will indicate the reverse file. Okay, clear? The single end sequencing and the paired end sequencing. Now next term is the depth. Any idea about the depth? Kansha ma'am, if you know anything.
No, ma'am.
Okay. What about others? Have you ever heard about this depth word? So depth means how many time the base is readable, or you can see the sequenced by the machine. That is what the depth. So for example, this is my reference genome. On this reference genome, what we are doing, we are aligning our data on this reference genome, right? So for example, on this particular position, there is adenine. And after mapping the reads on this reference genome, on this particular position, what we saw, it is having the adenine. So how many times the adenine it is readable here? Can you tell me? How many times?
6.
So you can see here the count 1, 2, 3, 4, 5, and 6, right? So 6 times. So depth here for this read would be what? 6x. So 6x is denoting what? The 6th time the adenine is readable by the machine. Got it? The depth word.
Yeah.
Because these terms you will get during the analysis. So that is why before the analysis I am just explaining the terms, because in the analysis we are not going to discuss this depth coverage. Okay, so that is why I'm clearing this particular depth word. What is that? Clear, all of you? Next is coverage. So coverage means how many reads are able to cover the reference genome. So for example, again, this is my reference genome and I am having a 5x depth. Okay. And that means here the reads are covering like this. So here you can see 50% of data is covered. Okay. 50% of your reference genome is covered and 50% of the reference genome is uncovered here. Correct. And here you are missing the data, right? Because we want to retrieve the complete information. Correct. So coverage means what? The how many times the reads are able to cover the reference genome. So this coverage and the depth, it is directly proportional to each other. How? If I'm increasing the depth, reads the sequence by the machine will be getting increased. So if I increase the depth here, so automatically the reads will cover the reference genome. Okay, and it will give you what? The 100% coverage here, because it is covering entire reference genome. So can we say, if we are increasing the depth, automatically the coverage is getting increased here, right? Clear? So what are the cutoff for the depth coverage? So like during analysis, I will tell you what we are, because for the germline and the somatic data is different. Okay, but the thing is, like, it is clear, the this part, the depth and the coverage. Any doubt?
No, madam.
Okay, next is the Fred quality score. So it is assigned to each and every base which is sequenced by the machine, and every base which is detected by the machine has some quality score, which will tell you the accuracy, that how accurately the base is called by the machine. So cutoff for this Fred quality score is greater than equal to 30. Why 30? Yeah. So can you see this table, Fred quality score table? So first column, it is giving you the Fred quality score. If my data it is having the Fred quality score is 10, okay, the probability of incorrect base call is 1 in 10. Here 1 in 10 means the base is sequenced or readable by the machine is 10 times. Okay, and out of 10 time, the one time the base was incorrect in my data, then base call accuracy is 90. For example, the Fred quality score is what? Sorry, 10. So 1 in 10 means the 10 time the base was read here. Okay, and out of 10 time, so for example, on this reference genome there is the adenine, and one time the base was here, the guanine. Okay, you can see the total 10 reads are here. Okay, and the 10 time the base was read, and out of 10 time the one time the base was incorrect. Here you can see there is the guanine instead of adenine. So that is what the quality score here. The base call accuracy is 90%. But if you increase the Fred quality score to 20, it is reading the base, that is the 100 time. And out of 100 time, the one time the base was incorrect, then accuracy is 99%. Now, if you again increase your Fred quality score to 30, or you are having the data to 30, you can see the 1000 time the base was read by the machine, and out of 1000 time, one time the base was incorrect. So it is giving you 99.9. So in the Word file, I've written cutoff is greater than equal to 30. Why? Because if you increase the Fred quality score to 40, 50, you can see the base call accuracy is only just 99.9, right? So none of it is giving you 100% efficiency. Can you see that? So that is why we are taking the cutoff for the Fred quality score is greater than equal to 30. Okay. So below that, you are not reading the data. Okay. Because for that, the confidence is low. Clear?
Yeah.
What about others? Is it clear? Um,
Yeah.
okay, fine. Next is BAM SAM file format. So these type of files, it will give you the complete alignment information, or you can say the mapping information. So that you are getting in the two files, that is the BAM, which stand for binary alignment map, which is a binary file and it is a computer readable file. Whereas this SAM file, it is a sequence alignment map and it is a human readable file. So these two files, it is having the same information. Okay, the mapping information you are getting in this form, BAM and the SAM. But whenever you are running the tools, so it requires the computer readable file, that is the binary file, because computer cannot read the human language. Okay, so that is why we are converting this SAM to BAM file and BAM to SAM file according to our useful. Okay, because if you want to read your file, that time you have to convert the BAM to SAM, right? Then only you can read. So that is why we were installing the SAM tools. SAM tools, it convert the BAM file to SAM or vice versa. Clear?
Yes, sir.
Next, you will get the PCF or VCF file. That is very important in this DNSSEC. So this is a variant calling format. Okay, the VCF file. Whereas this BCF stand for binary calling format. So whatever the you are calling the variants, so that you will get in this two file, in the BCF file and the VCF file. BCF file again will have the binary format of your variants information. But if you want to read your variants, like at what position might the change of nucleotide it is happening, okay, whether it is exon or whether any feature it is present, so that you will get this output, the VCF file. Okay, this file we are going to analyze. Okay, so this is the last step in the DNA-seq. Whereas this VCF file, it will give you three sections. That is, first is the metadata, second header you will get, then third you will get the data lines. So that we will discuss during the analysis only. Just for now, it is just divided into these three part: metadata, header, and the data lines. In the data lines, all the information of the nucleotide changes is present. Header means the title of the columns you will get. And in the metadata, like what type of tools with which VCF file you are generating, that tool information you will get in here in the metadata. Okay. What is a cigar string value? So it represent what, or you can say how bases from read is aligned to the reference genome. So for example, this is my reference genome. And there is one read, which is a 50 base pair, which is aligning on this reference genome. So cigar string will tell you how the B bases from the reads it is aligning on your reference genome. So for example, if this 50 base pair, okay, all the 50 bases are matching on this reference genome, the cigar string value will give you this 50M, where this M is what? Associated operator. So M stand for matching. So 50M will denote like the entire 50 bases, it is matched on your reference genome. But if you get this 40M, like this 40M10D. So that means out of this 50 base pair, only 40 bases are matching on this reference genome. Okay. And D, where this D stand for deletion. If you are having 40M, 5D, and 5I. So here that means 40 base pair is matching, 5 base are deleted, and 5 bases are inserted. Okay, so these are cigar string value. Got it? Any doubt?
No, madam.
Okay. Actually, the mapping quality and the Fred quality score is almost same. But here the Fred quality score, here the for the quality of the base is calculated. And here in the mapping quality, the read quality is checked. Okay, on your reference genome. So for example, this is my reference genome. The reads are mapped on the reference genome here. So this is the 30%. So you can see out of this 3 read. So this third read, okay, the one which is having the 90%, which is telling you what the more number of bases from this read are matching on your reference genome. So here for this particular read, the mapping quality is good, right? If you compare with this respect to this, like A and the B read, right? So mapping quality, it is telling you how the read is aligned to your reference genome, and it will give you the probability of the mapped read. Okay, whether it is has aligned at the correct position or not. Got it? Done? Okay. So let's see how to download the data from the NCBI SRA. Now, for example, our application name is what? The DNA sake. So I will just write here DNA sake, and I want to analyze the variants, right? So what I will just type variant calling. So it's up to you. You can write the cancer also. So if you want to use the operator, it will be specific for that. Okay. So just open that. Now you can see these many hits you are getting for this. These are total count. Uh, okay. Now on this left hand side, you can see assess here. So these are divided into control and the public. So in the control, what is that control means? So the data which is present here in the control section, that means you cannot freely access that data. So for that, you have to take the permission from the owner in the controlled access. But wherever, like you can see the public, so in the public what happened, it is a free of cost, so you can download the data in your system also, and you have the access for that particular data in the public. Source is the DNA here, type is the exome. If you want to do the whole exome sequencing, you can just select the exome from here and you can work on that. The library layout is the single end and the paired end. It's up to you again. If you want to work on single end, you will just have to select that single end. It will give you the hits for the single end only. Then again, the platform, it's up to you on which platform you want to work. You can just select that particular platform and it will give you the data for that specific. Then again, strategy is exam, and here the data in the NCBI SRA, it is also stored in the clouds. And the file type you will get here in the FASTQ format and also in the BAM format. Now, whenever you are downloading the data from the NCBI SRA, never go with this first page, okay, first second page. Why? Because still the research is going on and it is not updated till yet. So I would suggest whenever you are downloading the data from the NCBI SRA, you have to go with the oldest entry, okay. Now, for example, we can open any of this data. You can see here, I will just tell you later on like what the data which we are going to download in your system. So you are not going to download this. I'm just giving you the brief description for now. Okay. So this is just a title of this DNA sake of human. They have taken the human sample. Illumina machine they have used. How many spots have been used in the Illumina? You can see that. The total size of your data. Then design. So they have used the Illumina next sequencing machine, and this kit they have used. Then who have submitted the data? Then study is to precise the variant calling integration aid tools. If you want to read more about this article, you can just open that abstract and you will get an idea for this particular data. Then these are sample, organism, and here you can see the single layout, right? So how to download this single end data? You just have to select this run entry. And you can see the FASTQ download here. And you can download the data from here, okay, from the NCBI SRA. But it's just for the single end data. You can simply download from here. But what if my data is paired end? So that time I told you, you are getting two files, that is the forward and the reverse file, right? So if I select the paired end entry from here and I will open this, just let me check whether, yeah, this is the paired end data, right? If I want to download the data, just open again from this run entry. This is a FASTQ. I guess it is not here. Present. Yeah, just select this FASTQ format. So you can see by selecting this FASTQ format, you will get a file. Okay. So it will not separate your forward and the reverse file, right? So this is one of the drawback here for the paired end sequencing. Whenever you are downloading the data, never go with the NCBI SRA. Okay. Because it will generate only one file. It will keep both the single end and the paired end data together, okay, there. So that time you have to just copy this SRA ID and go to one of the database, that is the ENA. And then put that ID. So same information you will get, human, paired and Illumina platform. Below you can see the two IDs are separated. Can you see? So underscore one is your forward file, underscore is your reverse file. Can you see? This is the paired and data. So these two files you cannot see here in the NCBI SRA, right? So I would suggest whenever you are downloading the single end data, you can go with the NCBI SRA. And when you are having the paired end data, that time you have to get the session ID that you can load here in the ENA database, and you will get the two separate files. Got it? Now, I will share one link with you. Just open this in your system, because for this particular data, we are going to download. So everyone open this data. So we are going to run the DNS-seq pipeline on this particular data. Did you got this link?
Yes, ma'am.
Okay. Now for this data, you can see again the information Illumina MI-seq paired end sequencing. So this type of data is what the paired end, and they are using the targeting sequencing of 42 primary medulloblastoma sample using this Illumina TrueSight tumor 15 assay. So this experiment setup involved a specific NGS approach to study the medulloblastoma, which is a type of brain tumor. So here you can see the TrueSight tumor 15 assay. This is the targeted sequencing panel. So this is a panel which is designed to capture specific region of interest in the genome. And this 42 primary that indicate the number of tumor sample being analyzed. And you can see the title here, submitted by this department. Now, if you want to study more about this article, you can see here. The mutation status of TP53 was analyzed by this NGS using this particular machine. This is the sequencing machine platform they have used, where they are going to analyze the TP53. Why TP53? Because TP53 is one of the gene which is a tumor suppressing gene that plays an important role in preventing the cancer mutation. Okay, or you can say the in preventing the cancer. If there is a mutation in this TP53, it can lead to the cell growth which is uncontrolled and the tumor formation. Got it? Why we are going to analyze this TP53? Because if there is any mutation here in this TP53, it will create a cell growth, which is further leading to the tumor. So it is used to analyze the TP53 gene here in this tumor sample. Then they have done the DNA extraction. Then after DNA extraction, they are doing the quantification, because you have to see the amount of the DNA which is you are going to measure, because it should have the sufficient amount of the DNA. Then only you can proceed further, right? In the wet lab. Then after quantification, you are doing the library preparation that we have discussed in the yesterday session also. We are doing the fragmentation. Then we are doing the adapter ligation. So here, this is the targeting sequencing panel, right? So here, what you can do, you can design the primer for this TP53 also, okay? Because here, you already know what you are going to analyze in this sequencing. Okay, so that primer you can ready or you can prepare in your labs and you can insert. Then after adapter ligation or the primer, you are doing the library amplification. Then sequencing you are performing in this particular platform. Okay. Then again, the tools they are using to identify the genetic variation such as the SNP, Indel. So you can see the Lumina platform. This is the whole exome sequencing, PCR, paired end. Now, how to download the data, I will tell you. So all of you open this in your system and you have to copy this ID. Go to the ENA browser, then paste this. Then you can match your data, Illumina, MySeq, genomic, paired, whole exome sequencing. And you are getting this two file, forward and the reverse. Okay. Now, what you guys can do, just create one folder with the name of DNA sec. Remember that whenever you are creating any folder, don't give any space in between. Otherwise, tool won't run. Now, after this, what you have to do, just open the WSL. Now type here touch and write DNA sec pipeline. Enter. Then LS. So LS means how to check that whatever the files or the folder that is present here in the DNA sec. So LS stand for list. So you can see the pipeline is created in your folder, and touch stand for if you want to create any file in your system, you can use the touch command and just write the name of that file. If I go inside my folder, you can see it is created. Now open that in the notepad. Now just write here data selection. Sorry. paste that link. Then download the data is your step 1. Now, how to download the data? Just write here wget -c, and after -c, we are going to ENA browser. So this is my forward and the reverse, right? So you just have to copy the link address from here, go to the notepad and paste here. Same thing you have to do for the reverse also. Just write here wget -c, and then from here copy the link address and paste that. Are you guys on the same page? So now I will tell you how to download the file from the Linux. Okay. So it's up to you. If you want to download from here also, you can download. But sometime what happen, it is taking a huge time in the system. So that is why I want to avoid this. So I am preferring what? The Linux platform. So in the Linux, what we are doing, we are downloading the files or the folder with the help of wget -c, and then give your FTP link of your file. So now what I will do, I will just copy this command, and remember, wherever the folder you have created, from here you have to open the WSL and run this command. So you can see it is downloading the forward file.
Hello, excuse me.
Yeah.
Yeah, the first link, na, like which link we need to copy? Can you open the notepad?
This, sir. This one.
Yeah, please. Yeah. First one, data selection.
This one.
The first link. Yes.
Okay. I have
which-
already shared in the chat box, sir.
Oh, it is not. We are copying from the website, correct?
No.
See,
See,
remaining two things we used to get from website directly, correct?
exactly. So the link which I have shared, it is just to tell you how, from which article I'm getting this data, sir.
Oh, okay.
Okay.
It doesn't have any. Okay, only for week it is useful for here.
Yeah. So this, if I open in my browser, it will open this particular data.
Correct? Okay. Got
Okay.
it. Yeah. Thank you.
Now, if I write here ls, you can see it is downloaded. It is in the compressed format. Now, same thing you have to do for your reverse file also. Copy that and then paste here in your terminal. It will download the reverse file. Did you got it? How to download this file? Any doubt till here? Till the data downloading? So you can see the raw data which you are downloading, it is in the FASTQ format. Can you see?
Uh, Sorry to interrupt. So
Yeah.
how do we can execute this pipeline command? We have to put dot front slash. We need to give the DNA sequencing, or what is the command for executing? The notepad you have written two commands, correct?
This one, sir.
Yeah, yes. How do you execute this file from the terminal?
Yeah. So see, you have to just copy this command, wget this. Then,
Oh, okay.
yeah, you have to just create one folder with the name of DNSSEC, and from here
Yes.
you have to open the WSL, sir.
Okay. Suppose if I want to use in terminal directly, can we execute as a batch file DNA sequence pipeline?
No.
Anyway, we have.
See, you can you cannot run directly. So what happen if you are running on different, for example,
Mhm.
you are running on this
Okay.
WSL, and you can run from anywhere. Okay, it will start downloading this. But what happened? It is downloading
Mhm.
here in this.
Mhm.
But I want my data should be in one folder only. Now I don't want any confusion, right?
Okay.
During my project work.
Okay.
That is why I told you to just create one folder with the name of DNSSEC. So inside that DNSSEC, will create the files. All of thing execution we will doing in the one folder, sir. Okay.
One basic question. Then, what is the need for creating the DNA sequencing pipeline? You using touch.
Okay.
You have
See
created.
this pipeline.
Yes.
Okay. So why? See, if you are not saving the command, sir, in future you are like you will forget the command also, na, sir.
Oh, it is not for automation purpose, correct? Like a script file you can execute directly. It will
No,
execute only one.
you can also use the automation also for the automation. Once the all the commands it is written here in the pipeline, you can execute that also, sir.
So how do you execute? Like it's simple execution file, like put dot front slash and put the name of the executables like this.
Yes, sir. How to execute that? I will tell you. For
Okay.
example, uh, so you have to create the touch, maybe the pipeline dot sh. Okay.
Okay.
So if you want to run this pipeline dot sh, so what you have to execute? You have to write sh here, then give the file name, sir.
Okay.
So whatever the commands that is written here in the pipeline, it will automatically run in the terminal, sir. But
Okay. So.
remember that, like whatever the commands it you are not going to run, just put their hashtag, okay, before that.
Okay.
And what are the
Okay.
command that you want to run in your script, don't put the anything, okay, hashtag before that the commands. So automatically,
Okay.
if you want to run automatic pipeline in your system, so it will take this too. Okay.
Okay. You got it. I
Yeah,
can say like, pipeline, whatever you are using is like master file. It will capture all the commands which you are using for this pipeline.
exactly. Yes, sir. Yes, sir.
Yeah. 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 Thank you.
Okay. Yeah. So you all of you guys, like you can download this data in your system, and then further in tomorrow session we are going to check the quality of our raw data. Okay, with the help of
Madam,
FastFC tool.
madam, hello.
Yeah.
Uh, how to install in the SAMS tool in Mac OS?
Okay. Oh, sorry. I just forgot. Like you also have to. You can just download the data, Abhishek.
Yes. And I
So
Mac OS.
yeah, there
But
also in the
I
Mac
am facing,
there is a
I
terminal,
am,
sir.
yes, terminal.
Terminal. Just open the terminal. Okay,
Yes.
maybe Abhishek sir, what you can do, I have a session right now and you can join tomorrow
Okay, fine.
before
Okay,
the session, maybe before 7, sir, so that I can execute in your system also.
okay.
Okay?
Fine,
Yeah.
fine. Okay.
Okay. Okay, then we'll meet tomorrow, guys.
Okay. And also I am sharing in my screen. Please solve this problem. I am request you.
Yeah. I will just ping me like Akansha ma'am did today.
Okay.
You can also
Okay.
ping me tomorrow in the group.
Okay. Okay. Thank you.
Yeah. Okay. Okay.